to put a link up, right?
Yep, just like I did last time. Allowing you not to forget all this information over the break. So awesome, right? So we started our discussion with psychopharmacology, right? That was this section of the class. Yes?
Yes.
And we went all the way to the senses. So we have psychopharmacology, research methodology, and the 5 senses. So let me get my review notes. I'll be right back. What do we call the study of, turn this on, sorry. Oh no, no battery pack. Oh no, sorry.
Really? Serious?
Yeah, there's no battery pack. Sorry. So I guess you're gonna have to take very careful notes. So sad.
You just need to rewrite the test paper.
No.
Yeah.
No. All right. So what is the study of psychopharmacology? What is it?
Study of drugs.
Yeah. So the study of drugs and what?
The effect.
The effect they have on human behavior, right? Yeah, it's the study of drugs and the effect they have on human behavior. That is our definition of psychopharmacology, right? And so what are some of the ways that drugs can impact you? So how do drugs work? Psychoactive drugs.
They alter your perception.
They do, but how do they do that? Magically?
They have high lipid solubility.
Yeah, so any psychoactive drug is going to have to be lipid soluble, right? Otherwise it can't get across what thing?
Blood-brain barrier.
The blood-brain barrier, sure. And once it gets to the brain, right? So there's different things that allow it to get to the brain faster versus slower. If it can get to the brain faster, you need less of the drug to have an effect. And also it impacts behavior much more quickly, right? So what are some fast ways of getting to the brain?
So you can
Intravenous.
Yeah.
directly into
Intracerebral is what that means, directly to the brain. Intravenously, right into a vein. Or intraventricularly, which is into a ventricle, right? Any of those directly into the brain. So what are some slower ways then?
No.
Orally, inhalation, intramuscularly, all of these are slower ways. And so faster means a more immediate effect on behavior, right? And less of the drug is needed. All right, but once it's there, how does it work? Okay, I've made it to the brain. I'm a lipid-soluble drug. I have made it to the brain. How am I going to do my thing? So I heard the word neurotransmitter. That makes me happy. Yes, so neurotransmitter. It does something to neurotransmitter. Yes.
either it helps the neuron fire or stop it from firing.
I would never characterize it that way ever.
Agonist.
You're close. Yes, but that's not about firing versus not firing. Because you can have GABA agonists, which make neurons less likely to fire. Yes.
It helps or stops neurotransmitters?
Yeah, so it helps or inhibits the neurotransmitter's job, whatever that job is, right? And let's say, so if you have GABA is a neurotransmitter that does inhibition, right? Definitely. So I would definitely know that all the different neurotransmitters and what they do. That would be a really good place to start, right? like GABA, glutamate, acetylcholine, epinephrine, norepinephrine, dopamine, and serotonin, and maybe histamine and adenosine. We talk about those a tiny bit. But those 7 for sure, you need to know what they do. What do those neurotransmitters do? So GABA is 80% of your neurons use which 2 types of neurotransmitters? You remember? 80% use the same 2 neurotransmitters.
Glutamate and GABA.
Yeah, glutamate and GABA. The basic excitatory neurotransmitter is what? Glutamate. The basic inhibitory neurotransmitter is what? GABA, that's right. And so if you have a GABA agonist, that's a drug that helps GABA do its job. Like Valium. And Valium is a sedative, right? It causes you to become sedated because it increases the effects of GABA. And so we classify drugs as either agonists or antagonists, depending on whether or not they help the neurotransmitter do its job or stop it from doing its job. And that it has a very specific academic phrase that we use to describe that idea. It's called, what does the drug do to the postsynaptic effects? Postsynaptic effects, because there's a listening neuron, right? That listening neuron is the postsynaptic membrane. And so we ask the question, does this drug increase the postsynaptic effects of this neurotransmitter? If it increases those effects, it's an agonist. Does it decrease the postsynaptic effects? Then it's an antagonist. Make sense? So you can have an agonist that actually makes neurons less likely to fire, right?
Makes sense.
So that's important to be able to distinguish. And you should. So there are a lot of drugs we went over in class, right? And I think it is very fair for all the drugs we went over in class to know whether or not they're antagonists or agonists and what neurotransmitter they target. I think that's a fair thing for you to know. But we gave a lot of detailed information about the drugs, right? And all 30 drugs we went over, I don't need you to memorize every single thing about them. But you might want to take a handful of them and know them really, really well. Be able to describe them very, very in-depth, a handful of them, theoretically. But for all of them, you should at least know whether or not they're agonists or antagonists and which neurotransmitter they target. So you can give me an example of one of the drugs we went over in class. Methylene dioxymethamphetamine. And you should know the proper names for things. Right? So if you choose to talk about the street drug ecstasy, you should know that it's properly called methylene dioxymethamphetamine, MDMA. That is the chemical name for that particular drug, right? Or lysergic acid, LSD. Right? And so let's talk, so if you were going to describe, so if you wanted the basic knowledge about these drugs, both MDMA and LSD target the same neurotransmitter. Which one?
Serotonin.
Serotonin. Are they agonists or antagonists? Do they help serotonin or stop it from doing its job?
What's that stuff?
Agonist, which is why they cause euphoria. Because serotonin regulates mood. And you should know that serotonin regulates mood. And so if you were going to be talking about LSD or MDMA, and you say, and then knowing that they're agonists for serotonin would give you a lot of information about how they might alter behavior. They both cause euphoria. They do. LSD and MDMA both cause feelings of well-being and euphoria, happiness. But they have different sites of action. Now, you don't have to memorize the sites of action for all 30 drugs, but you should probably, as I said, take a handful of them and know them in great detail. So let's talk about the site of action for LSD is that it stimulates serotonin receptor sites. That's what it does. Those listening parts of a neuron that take in serotonin, LSD stimulates them all over the brain. So it also affects appetite. It also affects visual perception. It also affects thinking processes, cognition. MDMA has a different site of action. It's actually got two. It's still an agonist, but it doesn't necessarily stimulate receptors. Rather, it causes serotonin to be released from the vesicles. That's one side of action. It also prevents reuptake. So it spits out serotonin and then keeps it there. Because what is reuptake? Right, so remember, terminal buttons release neurotransmitter. You remember that? And then reuptake is that it sucks some of it back. So there's less of it in the synapse. So any drug that prevents reuptake has got to be a what? An agonist or an antagonist? An agonist. You have to actually think on the exam. You can't look for the word prevent and think to yourself, oh, must be an antagonist. No, because if it prevents reuptake, do you have more or less of the neurotransmitter? It prevents reuptake, because what is reuptake? It's the sucking back. If you prevent the sucking back, do you have more or less in the synapse? More. So any drug that prevents reuptake is an agonist. Prozac was one of the other drugs we learned prevented reuptake of serotonin. So it is also a serotonin agonist. Methylphenidate it doesn't target serotonin, but rather it targets dopamine. But it also prevents reuptake. Methylphenidate prevents the reuptake of dopamine, used to treat ADHD. Right? So know two or three or more, a handful of the drugs, know them pretty darn well. Could be, tell me how they affect behavior. Any specific sites of action. But then for all the drugs you went over, know which neurotransmitter they target, know whether it's an agonist or an antagonist. So what are all the different sites of action we talked about? What are the ways that a drug can change neurotransmission? Well, preventing reuptake is one of them, right? That would be an agonist. Causing neurotransmitters to be released, that would be an agonist. That's an example of a site of action. Yeah.
Enzymatic deactivation.
You can either prevent or encourage enzymatic deactivation. That's right. So if you prevent enzymatic deactivation, are you an agonist or an antagonist? Yeah, you're an agonist. Just like if you prevent reuptake, if you prevent enzymatic deactivation, it's also going to be an agonist. Yes.
When the enzymatic deactivation, the neurotransmitter is released and it's not deactivated. Does it hold it all right there in the synapse, or is it
generally it is enzymatic deactivation that ensures neurotransmitters stay at the synapse.
But then if it stops, does it just?
So if you prevent enzymatic deactivation, then yes, you should have that neurotransmitter start to spread and affect other neurons that have those receptor sites. So how else? How else can a drug impact? What kind of sites of action are there? We can stimulate receptor sites. Okay, so receptor sites themselves. Aren't there some drugs that actually interact with receptor sites directly? Yeah, there are. And in class we talked about the difference between competitive and non-competitive binding. Because some drugs bind directly to receptor sites. Do you remember that? And and that could be an agonist process or an antagonist process, right? Because the drug could help ion channels open. And if they're helping sodium ion channels open, or not sodium rather, but if they're helping the ion channels open, that's an agonist. If they're stopping the ion channels from opening, they're an antagonist. So what's the difference between competitive and non-competitive binding? What is the difference? So there's two different ways that drugs can bind. They can do so competitively or non-competitively. Who remembers our discussion of that? Yes?
Competitive is when, so whatever it's the thingy,
The receptor?
Yeah, the receptor.
When the neurotransmitter hits the receptor and
Yeah.
binds to it?
Yeah.
Yeah?
If the drug or something, whatever hits there first is what works.
And so what would be that? So the drug gets there first, and then the neurotransmitter can't get in now. What's that called? Competitive.
Yeah.
I like to think of them as like siblings, right? So you've got drugs that target receptor sites are like siblings for neurotransmitters, right? And they come in two varieties. The competitive kind, where the drug gets there, is like, ha ha, I'm here. Ha ha ha. You can't get in here now, right? Versus non-competitive binding, where now they sit right next to each other, right? And sometimes that can be a helpful relationship. Agonists, like the drug and the neurotransmitter are conspiring back there, like siblings might in the back seat, right? Or it can be antagonist, non-competitive binding, where they're like fighting, where the drug is trying to interfere with the neurotransmitter. So non-competitive binding means that both of them are there in their own different binding sites, and then the drug does whatever effect it's going to have, agonist or antagonist, non-competitive. They both can get into a receptor site. Competitive binding is that the drug is there, and so the neurotransmitter can't get in. It's blocked.
So, would there be too many neurotransmitters for how many binding sites there are? It's just
No. So for example, because you're on a particular, there's a particular receptor site, right? And we don't even learn about all different kinds of receptors, right? Your book talks a little bit about it, right? They get these cool like Greek letters and stuff. And there are dozens of different types of receptors. We only focused on like a couple of different varieties, right? And so essentially there are some receptor sites, some types of neurons, some types of processes, where there are multiple different types of binding sites. And so you can end up with non-competitive binding, where there can be multiple binding sites of the same receptor, where the drug can get in there as well as the neurotransmitter. The drug being there does not prevent the neurotransmitter, so it's non-competitive. But there are other processes and other types of receptors that once the drug is in there, there's like no vacancy signs. Makes sense? Competitive binding. So, but really, any aspect of neurotransmitter's existence can be altered by a drug, right? There are drugs that increase production, decrease production. There are drugs that fill up vesicles, so they're like bursting at the seams. Other drugs that empty out the vesicles. Drugs that cause the release. Other drugs that prevent the release. Drugs that increase reuptake. Other drugs that prevent reuptake. Drugs that stimulate receptors. Drugs that inhibit receptors. Drugs that block receptors. Drugs that help, right? And not blood, drugs, right? So pretty much any aspect of a neurotransmitter's life cycle can be altered, thus altering the postsynaptic effects of that neurotransmitter, right? And then there were three drugs that we sort of talked about that were in a class by themselves that didn't make a whole lot of sense to talk about as being a particular agonist or an antagonist for a particular neurotransmitter. What were those three?
Opiates.
Opiates, that's right. Heroin, morphine, codeine, opium. All of these are opiates. And you can sort of kind of think of them as agonists because they act just like a naturally occurring chemical in the brain called endorphins. And chemicals that are very similar. There's actually a bunch of different chemicals that do the same sorts of thing. The one you've probably heard of is an endorphin. And an endorphin does interact with many different types of receptors, and it is a neurochemical, but we don't think of it as like a direct neurotransmitter like serotonin. Rather, endorphins interact with neurons, trigger things like the pleasure centers of the brain, trigger the relaxation centers of the brain, help with pain reduction. Endorphins trigger these circuits. And so opiates act as if they are endorphins. And so you can sort of kind of think of them as an endorphin agonist. But really, it's because they're acting as if they are an opioid, an endogenous opioid. Same thing with what other drug?
THC.
THC, the active ingredient in marijuana. The active ingredient in marijuana interacts with the receptor sites for naturally occurring endogenous cannabinoids. you also have chemicals in the brain called cannabinoids that interact with a whole lot of different types of receptor sites and a whole lot of different kinds of neurons and target lots of different circuits like appetite circuits, memory circuits, time perception circuits. And so THC acts as if it's an endogenous cannabinoid. So you can sort of kind of think of it as an agonist for cannabinoids, but that's because it acts as if it's a cannabinoid, which is a chemical that interacts with lots of different brain circuits. And the last one we talked about that's in a class by itself is what?
Alcohol.
Alcohol! Because it doesn't target just one neurotransmitter, it targets a lot of them. It's a glutamate antagonist, a GABA agonist, an acetylcholine agonist, a norepinephrine agonist. It just gets all a bunch of stuff going. Right? So it has a very profound effect on behavior, a whole host of different systems. And so if I was going to learn some drugs in a very detailed way, I would be cautious selecting alcohol, THC, or opioids because the level of detail required to talk about these three drugs is a whole lot higher, right? But you should certainly know how those three drugs work. Make sense? You could talk about those theoretically if that showed up in your future life. The level of sophistication for those three would be higher, right? All right. So know about the drugs.
Something tells me you're going to want a short essay answer.
All right. Moving on.
Choose a handful of drugs and write about it. Okay.
All right. So then we talked about research methodology. About research methodology. And we talked about different sort of types of research methodology, right? We talked about, so what's the biggest, most important type of research methodology? Probably the most commonly used. And one we talked about first.
Lesion studies?
Yeah, ablation studies, lesion studies, experimental ablation. And the purpose of this type of study is to destroy a part of an animal's brain and then see how that affects behavior, right? That's sort of the first step. Use what? What do we do in order to do an ablation study? What is that called?
What?
How do we do an ablation study? Do we just like stick a knife into the animal's
slice.
brain and we just like poke at it and go, do I start there?
You preserve it?
Pardon?
Preserve it?
Well, what do I have to do first? So I have a live animal. He's running around. I'm all, Mr. Whiskers. What am I going to do to Mr. Whiskers?
Kill it.
First?
Fixation.
Observe.
Damage a part of the brain?
Stereotactic surgery, maybe? Get him to hold real still so I can actually destroy the part of the brain that I want to destroy. Create the lesion. A lesion just means a site of damage. That's what I probably should do first. Stereotactic surgery, create a site of lesion, and then watch how the animal's behavior changes. So I'm altering normal behavior. I'm changing it by destroying a particular part of the brain. Then the second step, which you all were getting at, are a group of methods called the histological methods. The histological methods comes next. First the ablation part of it, then the histological part of it. So yes, now we have destroyed the part of the brain, watched how the animal behaves. Now comes the histological methods. And the first step there is what? Fixation, preservation of the brain. How do we preserve this rotting piece of flesh? How do we preserve it?
Liquid formaldehyde.
Liquid formaldehyde, which gets a fancy name, is called formalin. Liquid formaldehyde, or another name for that is formalin. So yes, okay, we preserved the brain, great. It's preserved chemically, it's not gonna rot away on us. Now what do we do to it? Section or slice it, that is correct. How do we section or slice it? What machine do we use? It's called a microtome. A deli slicer is probably not the right answer on the exam. The word is microtome. And yes, it does create these very, very paper-thin slices of brain. Microtome. Now what do we do with it? Now we've got these paper-thin slices.
Microscope.
Well, if I shoved it under the microscope now, I'd see a big pink gray blob.
Stain it.
You need to dye or stain it. Two different common types of dye are called methylene blue and cressyl violet dye. That dye is the DNA of the neuron itself, called the Nissl substance, actually puts all the dye into the cytoplasm. So then you can see the protein structures around the cytoplasm. So the third step is dyeing. These are called the histological methods. Wow, knowing their proper name might be important. Histological methodology. Fix it, slice it, stain it. Well, how do you fix it? With formalin. How do you slice it? With a microtome. How do you stain it? Use the Nissl substance to put the dye into the cytoplasm. But then there's other ways of studying behavior, right? Don't we sometimes just explore the normal behavior of animals or people? Somehow measure, go, hey, just behave normally, pretend I'm not here, right? And then there's different sort of ways of doing this. We actually talk about lots of different research methodologies that essentially explore what part of the brains become activated when animals or people do normal stuff. Lots of different methodologies that do that. Remember what they are?
PET scan. Right?
PET scans is an example of a methodology that goes, okay, person, do something normal, and I'm going to watch, and I'm going to see which parts of your brain become activated. And positronic emissions tomography, a PET scan, the fancy name for it, positronic emission tomography, is cool because we actually measure how much glucose neurons are eating. Because we hook up glucose to a radioactive tracer and actually measure the gamma rays that get kicked off by this tracer. Positronic emission tomography, PET scan. But the goal of that is, here, clench and unclench your fist. I'm going to watch to see which brain areas get activated. That's what a PET scan does. There's other methodologies that have the exact same goal. What are they? An fMRI measures blood flow, right? Because parts of the brain that are more activated when a person is clenching and unclenching their fist or thinking about a particular memory, more activation means more blood flow. And so an fMRI is a giant magnet, a functional magnetic resonance imaging technique, uses a giant magnet not to measure magnetism, but to measure blood flow. That's what an fMRI does. It measures blood flow. But the goal is the same. Do something normal, and I'm going to see which brain areas get activated. There's other ones. And you can do, which one?
EEG?
Yes, an EEG. We do that to animals, right? We implant electrodes directly into an animal's brain, have that animal run around, and it's really, you can do it without causing brain damage, right? At least any profound brain damage. And you can measure the electrical activity of the neurons themselves. So again, we're seeing which part of the brains are more activated when the animal engages in normal behavior. But there are other research methodologies also, right? We have what are called the stimulation methods, right? Direct stimulations. So it's not looking to see which brain areas get activated when an animal engages in normal behavior. Rather, the stimulation methods are to like zap the brain and then watch how the animal's behavior changes. We talked about 3 different kinds of direct stimulation. There's electrical, which is like putting an electrical current through the animal's brain. Chemical, putting some excitatory or inhibitory chemical in the brain. And then the coolest, most sci-fi one ever, called what? Optogenetics. The optogenetic methods. Absolutely. What are those optogenetic methods?
When they do a light.
Yes, it does have to do with lights. What are the optogenetic methods of stimulation? Something out of sci-fi, but yet it totally is real.
They put a modified virus.
That's right. They take a genetically modified virus, and into the animal. That genetically modified virus goes to the brain, and what does that genetically modified virus do? Does it make the animal sick?
It creates new iron channels.
Yes, it creates brand new ion channels. You know those channels that allow a neuron to fire or not fire? You know those ones, those super important structures? So here's this genetically modified virus, modified by the researcher, goes into the brain and starts inputting new ion channels. Ion channel for you, ion channel for you, new ion channels all over the membrane of the neurons. And these ion channels are photosensitive, which means different colors of light open them or close them. Photosensitive ion channels. That way you can directly stimulate neurons or directly inhibit them using different colors of light. Of course, this is done on animals, not people.
I want to know what the colors.
I think the most common colors are red and blue, so don't worry about it. But I don't think we even covered that.
I guess that was last year.
All right. What other cool ways are there to study human behavior? Oh, we forgot one. We forgot one of the ones where a person engages in normal behavior, and then we see which part of the brain gets activated. Which one did we miss? fMRI, PET scan, EEG. A magnetoencephalography?
Yeah.
Is that the word you're looking for? Yeah. Now, this one often gets confused with an fMRI, but don't let it confuse you. A functional magnetic resonance imaging technique, an MRI, uses a magnet to measure blood flow, right? So, but the magnetism is the second word, functional magnetic. So that means it's not measuring magnetism, it's using a magnet. A magnetoencephalography actually measures the magnetic field put out by your brain when neurons fire together. A magnetoencephalography, using those things called squids. Little things all over your brain. Magnetoencephalography, okay, cool. And then we had the methodologies that looked for specific neurotransmitters or specific receptor sites. And we talked about a process, it's more like fine-tuned staining, right? It's much more fine than the kind of staining you would do in an ablation study. So what's another way we can dye the brain to be a lot more specific about the structures we're looking at?
Immunocytochemistry.
Immunocytochemistry, that's right. One of those other cool sci-fi ways of studying the brain. The goal, again, is in a dissected brain to stain the different structures. Immunocytochemistry does this in a super cool way. How does it do it? Is there a tiny little microscopic person painting the neurons? No.
Does it light them up?
It does light them up because it dyes them with fluorescent dyes, but how does it do it? Can you just dip the neuron into a fluorescent dye and you're like, it's good? How does it do it? How do you only stain, for example, the axons of a neuron? Or only stain like a particular enzyme of a neuron?
The antibodies target the specific proteins?
That's right, because that's how antibodies work. That is how your immune system works. So your immune system creates these things, these fighters called antibodies. And these antibodies wrap themselves around a particular protein. So let's say the axons of your peripheral nervous system, as an example, have a particular protein signature. So a researcher then might put a genetically modified antibody into that, or present it to that neuron, and now that antibody wraps itself around the axon, lighting it up with the fluorescent dye. Immunocytochemistry, using the immune system, antibodies, to chemically dye different protein structures. Immunocytochemistry. Yes?
So the antibodies are not destroying the neuron at that point?
Well, the animal is already dead.
Oh, that's right.
Yeah. Yeah. So you think of there's a slice of brain matter, and what they're doing is presenting this modified antibody onto the neuron, the tissue, and that then stains only the protein that antibody is intended to target. So yes, theoretically it would kill the neuron, but it's already dead, so it's fine. So know those, and then we also talked about the genetic methods, right? Which is a totally different goal. The genetic methodology. So we talked about twin studies, adoption studies, and direct manipulation would create the creation of knockout genes. So tell me about twin studies. How do they study twins to determine to what extent biology explains behavior?
DNA?
Sure, they compare the DNA, but do they grab do they actually take like a skin scrape of the twins and shove it under a microscope? Is that how they do it? No.
They said the concordance rate?
That's right. They compare these things called concordance rates. They compare identical twins, monozygotic twins, to dizygotic twins, and they compare the concordance rates. What does that mean? What is a concordance rate?
If the twins share the trait of concordance?
That's right. So if the two twins have the same trait, we call it concordant. And if identical twins have a high concordance rate for a trait, higher than non-identical twins, that argues for what?
Discordant. Oh wait.
If identical twins have a higher concordance rate than non-identical twins, monozygotic twins have a higher concordance rate than dizygotic twins, what does that suggest about that trait? That is a larger genetic component. A large genetic component, or at least has a genetic component. All right. So then we talked about the senses, right? We started with vision. And for all of the senses, I was real careful to track for you the pathway of sensation, right? The pathway of information, starting at the stimulus. Like vision, it's light, visible light is the stimulus. And track it all the way to the end of its journey, which for vision is one of two places. What is the end of the journey for vision? Pardon?
Optic.
That would not be the end of the journey for vision. What is the last stop? There's two stops actually.
Temporal and parietal lobe?
Yeah, but we call those something very specific when we're talking about vision. We call them two different streams. What are they?
Ventral and dorsal?
Yeah, there's the dorsal stream of information. That's the last stop. Or the ventral stream, also another. So there's like two last stops for vision. Now the dorsal stream, what kind of visual information goes up the dorsal stream to the parietal lobe? What kind of visual information?
Where are you in space?
Well, no. Where are those other things out there in space? Right? Like if I'm looking at the bee and I'm tracking where the bee is, the information about where that little guy is, where is that bee, is going upstream to the dorsal stream to the parietal lobe. That would be the last stop for where something is. Okay, so what about the ventral stream? What visual information is going down the ventral stream to the temporal lobe? Perception of what, though? Form. Yeah, form. How do I know it's a bee and not a flying mongoose? I don't know, whatever, right? How do I know it's a bee? I know where it is. Thank you, parietal lobe. Thank you, dorsal stream. I know where it is. But if I want to know what that thing is flying around, I'm going to need that visual information to go to the temporal lobe, where memories happen and they're stored and are created. What it is, what something is being processed at the last stop of vision in the temporal lobe, the ventral stream of analysis. But there's a whole lot of stops in between, right? Right? So for all five senses, we did those various stops, right? And there's a lot of similarity between the different senses, except for your sense of smell. All 4 senses have one stop in common. What is it?
The thalamus.
The thalamus, that's right. And there's different parts of the thalamus, like it's the LGN for vision, and the medial geniculate nucleus for audition. Sorry. Guess nobody was late today. It's amazing. You? I walked in exactly at 9:35. Like I do every day. All right. So we have, so that's going to be a very similar stop if you were going to be describing it, right? Yeah? Thalamus. So let's just track vision. So what is the first part of the nervous system to interact with visible light? The rods and cones. What would be step 2? Bipolar cells. That's right. Bipolar cells take information from the rods and cones. And then what would be step 3? The rods and cones are connected to the bipolar cells. The bipolar cells are connected to the ganglion cells. That's right. And it's the axons of the ganglion cells that create what?
Optic nerve.
The optic nerve. So I would say step 4 would be the optic nerves, right? And then I would, if I was going to be describing this at some point in my near future, I might say that the optic nerves cross where? Where do the left optic nerve and the right optic nerve cross? They go back into the brain and they cross somewhere. What do we call that place?
Optic chiasm?
The optic chiasm, that is correct. And now we have arrived, after they've crossed at the optic chiasm, what part of the brain have we now arrived at?
Striatal cortex.
Nope, first we have to go through the thalamus. The thalamus, right? We have to go through the thalamus, and then from the thalamus, what part of the thalamus? The LGN, lateral geniculate nucleus. Now we get to the striate cortex, which is in the occipital lobe, right? Striate cortex is our new fancy name for primary visual cortex. And then from there, visual information goes one of 2 places. What's that? Those last stops again? Oh, that's right. It's the what streams?
Ventral and dorsal.
Ventral and dorsal streams. And now I will have fully described this visual stream of information, starting at the rods and cones, ending in either the ventral or the dorsal stream. See how I was able to do that? I was able to describe that.
No, that was the second essay question.
But I could also do that for audition, right? And there would be some similarities between vision and their sense of hearing, audition. So so transduction happens in the rods and cones. Rods and cones, what kind of receptors are they? They're a particular type of receptor. They're a photoreceptor. That's right, photosensitive receptor, a photoreceptor. Ah, but where does transduction happen in the ear? Oh, it does happen in the ear.
Cilia?
Yeah, the cilia of what kind of receptor?
The mechanic one.
They are mechanoreceptors. That is correct. The cilia, the very tops of those mechanoreceptors, bend back and forth. That's right. But is that where it all begins? No. What's the first part of the body to interact with sound?
The pinna.
The pinna. But that's not where transduction happens, right? No. So already we have some differences between vision and audition. The very first part of the body to interact with visible light is actually the thing doing transduction, right? The rods and cones. But for sound, it's a little different because sound waves have to be amplified before they can get to the site of transduction. Transduction is just how we say that physical stimuli is translated, right? So first the pinna, and then what? This outside part here is called the pinna.
Eardrum.
Yeah, the eardrum or the tympanic membrane. Right? That's the second place on the brain, not in the brain, in the nervous system though, that interacts with sound. First the pinna, then the tympanic membrane. What's the third structure? The ossicles, because the tympanic membrane is connected to the ossicles. They're the malleus, incus, and stapes, but they're collectively the ossicles, the three tiniest bones in the human body, which amplify the sound. The ossicles are connected to what? The cochlea, which is like a curled up snail shell. And it is inside this fluid-filled cochlea that a membrane called the basilar membrane lives, and it is on the basilar membrane that the cilia live. These cilia of these mechanoreceptors are where transduction happens. Okay, so now we've got these cilia bending back and forth. Finally, transduction, hooray. Cilia bending back and forth. And these cilia are connected to what? I'll give you a hint, it's a nerve.
Auditory.
The auditory nerve. Yeah, the axons of these cilia, these mechanoreceptors, these collective create these bundles of axons that then bundle together to create the auditory nerve. And unlike vision, where the optic nerve goes straight to the thalamus, no, that's not the way it works for audition, for the sense of hearing. That auditory nerve has to go through the hindbrain, through the midbrain, and now to the thalamus. There are particular parts of the hindbrain and midbrain, but clearly that must not be what I think is the most important, because I'm not spending a great deal of time talking about it. And now from the thalamus, where do you suppose I'm going to go if I'm auditory information? The primary auditory cortex. But then from there, which is called the core region now, and then from there we go to the association cortex, the association area, which is called either the belt or the parabelt region. That's the last stop. And what information, what you hear, is processed on the left, and where a sound is processed on the right. So you hear how I've now described the sensory pathway for audition? Now if you look carefully through your notes, you could theoretically do that for all 5 senses. Do you imagine that knowing about all 5 senses is probably useful and important for the entire test? Probably. Do you imagine you're going to have to write long essays about all five senses? Probably not. But knowing the sensory pathway for all 5 would be very useful. You might want to know and be able to describe the sensory pathway with sufficient detail. But probably not for all 5, I'm guessing. So, tell me about the neurons of vision. So the neurons that live in your striate cortex, are they all identical to each other? Totally the same, no different from one another? That's just, they all do the exact same thing. Responsible for the same kind of information, responsible for the same portion of the visual field? No, how do they work? If you were going to describe the neurons of the occipital lobe, the neurons of the striate cortex, visual cortex, how would you describe them? What are some examples I gave of those neurons?
There's neurons that recognize position.
Yeah, there are uppy-downy neurons, sidey-widey neurons, vertical neurons, horizontal neurons. So they're highly specialized, right? And so very specialized, care about very different types of information, right? And they work in modules that share information, right? They're sharing information with each other. They work in these modules. This is what the research suggests. And do all the neurons, do they look at all the entire visual field? Or do they specialize? They specialize. I'm a neuron and I'm only responsible for right here. In fact, not only am I responsible for right here, I'm only responsible for firing if there's a vertical line. Very specialized. Working in modules that share information. So, why do we have a blind spot?
Where the optic nerve collects, there's no cones around.
Yeah, because the retina is backwards in organization, right? The rods and cones are in the back, and on top of those are the bipolar cells, and on top of those are the ganglion cells. But then the ganglion cells go back, cluster together to create the optic nerve. And so it's the placement of that optic nerve, where there are no rods and cones, because it's like the optic nerve is like, excuse me, rods and cones, I have to get through. There's no rods and cones there. So it's the placement of the optic nerve that creates the blind spot. So tell me about opponent process. So tell me about the rods and cones first. Tell me about the rods and cones. Which does what? Well, I mean, the cones respond to light information. Just when can cones work? What kind of light? Color and what else? What else do the cones do?
Specific shape.
Which one?
Specific shape.
Well, details. Very fine details. Right? So you need a lot of bright light in order for cones to work, right? A lot of bright light. Rods, they always work though, right? Now most of the cones are where? In the fovea, the center. Most of the rods are on the periphery. And these are photoreceptors. Photoreceptors. So when you wake up in the middle of the night, you're stumbling towards the bathroom, are you using your cones? No.
No.
Nope.
Rods.
You're using your rods. So tell me what opponent processing cells are. So ganglion cells are opponent processing. And many of the cells of your striate cortex are opponent processing. What does that mean? Cells that opponently process. There's a lot of different varieties of them. We talked about 6 different varieties. In fact, there are only 6 different varieties in the ganglion cells. More varieties in the striate cortex. What is an opponent processing cell?
It gave you the basic shape of the outline, the edges.
Well, on and off ganglion cells do that. On and off ganglion cells are examples of opponent processing, and it's how we code for edges. That's right. But what does it mean to say that they're opponent processing? This is what is called therapeutic silence. Well, what does the word opponent mean?
The guy you're playing against.
So that things are opposed. Who remembers our discussion of the visual system where there were things that were opposed to each other? So we talked about the visual system, neurons of the visual system. We talked about a certain type of cell where it seemed to be that. Yeah, and so what do I mean by opponent processing? How do they function? How do they work?
Some of it fires, another half doesn't, or
Yeah, you're definitely getting it. An opponent processing cell, there are some things that make it fire and other things that make it not fire. The exact same cell. Yeah.
So when something hits the center, it fires, and then if it hits the thing surrounding it, it gets inhibited.
That's right. So an opponent processing cell has these visual fields, right? The part of the visual field it's responsible for, and it's opponent processing if the exact same stimulus sometimes makes it fire and increases its firing rate, and sometimes decreases its firing rate. That is an opponent processing cell. And so let's say I tell you that this is this opponent processing cell, and when a green light hits the center of it, it gets an increase in firing. What happens when a green light hits the surround? Pardon? Yeah, there's a decrease in firing. You don't. All I have to tell you what happens to the center. Let's see, I tell you I'm an opponent processing cell, and when light hits the center of me, any light, I get super inhibited, the decrease in firing. What happens to me when that same light hits the surround portion of my receptive field? I get excited, I get increased. Opponent processing. I should definitely know about that. So pitch and how loud something is coded right at the little cilia, right? The cilia bend back and forth. Which cilia says the pitch of it is, and those cilia are arranged how on the basilar membrane?
Tonotopically.
Yes, tonotopically. That means the The high pitches are at one end and low pitches are at the other, right? They're arranged tonotopically along the basilar membrane. And that's where pitch is coded for, right? And also how loud it is. If the hairs get bent back and forth a lot, it's a very loud noise. It doesn't get bent very much, it's a soft noise. But how do we code for where something is? Where is that sound coming from? 2 ways.
Like the change in tone?
What do you mean by that?
Well, like, you know, the sound's coming from here, you know, this is getting a sound and this one's not, so you know it's coming from here.
Yeah, it's called the phase difference. Yeah, that's exactly right. The sound's coming from over here. The sound hits the left ear first and then the right ear, right? And this eardrum's getting pushed in and this one's getting pulled out. And this difference in phase, which phase did the sound hit each ear, is one of the ways we code for where a sound comes from. There's another way, much more detailed. Are these just for show?
Yeah.
How does the nature of the pinna allow us to code for where something is coming from?
How the sound bounces off and
That's right. That's right. Depending on where a sound comes from, the actual frequency of that wave gets a tiny amplified or a tiny bit diminished, depending, and there's a unique pattern of amplification and diminishing, getting smaller, for where it comes from. Where did that sound come from? So both of those ways are ways that we code for where a sound is coming from. So what 3 different senses live in our cutaneous sense, the sense of in our skin? There's 3 different senses of touch that live just in our skin. What are they?
temperature.
Temperature, coded by what type of receptor? Thermal receptors, yeah. What else?
Pressure.
Yeah, pressure and vibration, coded for by what kind of receptor? Yeah, low threshold mechanoreceptor, means it doesn't take very much for them to respond. Also called free nerve endings. Low threshold. And what's the third thing? Pain, coded for by something called the nociceptor. 2 different varieties of nociceptors. High threshold mechanoreceptors, as well as the ones that interact with things that burn. Okay, and pain, we also talked more about pain, right? So high threshold mechanoreceptors, but also where does the immediate consequence, like just your basic sensation of pain, what part of the brain goes, yep, that definitely hurts?
Hypothalamus.
So touch information, anything that says sense of touch goes to what part of the brain? Which cortex? So audition goes to the auditory cortex, vision goes to the visual cortex. Where does touch information go? Oh, I would totally know this. It's part of that circuit, yeah.
Is it somatosensory?
Yep, the somatosensory cortex. And in fact, where you sense pain is in the somatosensory cortex. That is where you sense pain. But do you experience pain as this neutral thing, no big deal? Or do you have an emotional response?
Emotional response.
You do have an emotional response. And so the second place where pain starts to get regulated, the emotions of pain get regulated in this place called the anterior cingulate cortex. But that's like the cussing that goes on with pain. Like, oh my gosh, this hurts. I'm upset. Where do you process the long-term emotional implications of pain? Like, what am I going to do about this pain? You're totally right. The prefrontal cortex is where you make a plan about pain. Like, oh my gosh, what does this pain mean? Am I paralyzed for life? Is this childbirth? Is this no big deal? Is this going to be gone in a few minutes? I'll just shake it off. Prefrontal cortex, long-term emotional implications of pain. And so the last thing we just sort of explored was the sense of taste and the sense of smell, right? Both called the chemical senses. We just went over this today. Why are they called the chemical senses? There's a few reasons why I might describe it that way.
Because they don't go to the, they don't go to the thalamus.
Well, that's true of the sense of smell, but not for your sense of taste. The sense of taste does go to the thalamus. Yes, it does. It goes, so the sensory pathway for taste starts in the taste buds, right? Different types of taste buds, is connected, come together to create the 9th and 10th cranial nerves. Those taste buds, the axons of them, are connected to bipolar cells. The bipolar cells are connected to the cranial nerves. The cranial nerves go in through the hindbrain, and from the hindbrain to the thalamus. This is taste, right? Gustation. And from the thalamus, it goes to this place called the gustatory cortex. Oh my gosh. I don't remember learning about the gustatory cortex in that first week of class. Because what other cortex is the gustatory cortex a part of? The somatosensory cortex, the tongue portion of the somatosensory cortex. But it's the actual chemicals of saltiness. Salt molecules interact with the tongue. A specific chemical. We can't taste everything, right? Only specific types of chemicals. And that's true of smell too. We can only smell certain types of chemicals. One of the ways they're called chemical senses. Another is the actual chemicals interact directly with those sensory neurons, right? Another reason they're called the chemical senses. But only olfaction doesn't use a thalamic relay. Only the sense of smell. Communicates directly. And so tell me a little bit more about the gustatory cortex. We talked about how the gustatory cortex is exactly the same from person to person or totally unique?
Unique.
Totally unique from person to person, right? My salty place is not the same place as your salty place, right? Both live on the gustatory cortex, which is a part of the somatosensory cortex, but it's unique from person to person. How many different types of taste receptors do we have again?
339.
Taste.
Oh, sorry.
How many different types of olfactory receptors do we have again?
339.
339. And yet, how many different things can we smell?
10,000.
10,000. Those are the parts of the information that I suggest you focus your attention on. And I promise you that only the things that came out of my mouth today will be on the exam, but those are certainly the places where you should focus your attention. Have a nice break. Don't forget about stuff.
We're getting great.